Hi everyone, thanks so much for joining me today. My name's Jacqueline Carville, and I work in marketing at DNASTAR, and so I'm here to introduce us briefly. So DNASTAR is a software company located in beautiful Madison, Wisconsin. We provide software applications for molecular biology workflows, protein workflows for structural biologists, and NGS workflows, which we'll be discussing more today. So the speaker we have here today is Matthew Kaiser. He's our Senior Manager of NGS Applications, and he'll be discussing our variant detection and validation control workflow, which is particularly useful for many people doing NGS workflows, also people doing clinical research as well. So with that, I'll go ahead and pass it over to Matt.
Okay. Thank you, Jackie, and thank you everyone for joining us this afternoon. Today we'll be talking about DNASTAR software and some of the newer workflows that we have in the software where we can validate gene panel and targeted resequencing data sets and use our software to determine accuracy of the entire process. So I'd like to show you an introduction to the company first, and then we'll take a look at some screenshots of the ease of use of the software as well as some of the results. So a little background to start out. DNASTAR is located in Madison, Wisconsin. There's a nice picture here of Lake Monona in the capital. We're just down the road from downtown Madison. Our developers, our sales team are mostly located in the Madison location. My role at the company is to work with customers on NGS projects. I listen to customer feedback, I relay that to the development team, and really spend a lot of my time in a support role. What's nice about having all of our folks located here at Madison is that I have direct access to the development team, so I can really relay information accurately. And I think that makes a big difference for developing top-notch software. So DNASTAR software is compatible with all the NGS platforms. Of course, Illumina and Ion Torrent. PacBio has been really an increasing presence, especially for microbial genome assembly. And we still do some 454 as well. So what's nice about this type of software is that it can accommodate all different platforms as well as all different types of workflows. And that can be important, especially for labs that are doing more than one thing or using more than one type of sequencing instrument. So one of the first questions that we often get is, what is the hardware requirements for this type of software? And this is a kind of a general overview of the type of hardware that you might use. It could be just a laptop computer. When we're doing alignments for gene panels or exomes, you do need to have enough space on a hard drive, and we call this a scratch disk over here. And the scratch disk processes its data as the assembly is running. And so for a gene panel, that might be 1 terabyte of space. For a whole exome, it might be maybe 2 terabytes and so forth. So it's really, this day and age, a fairly modest requirement. And of course, you need enough storage for the type of data that you have. So it's just something to keep in mind. Nowadays, most laptops have enough RAM, enough processing power to handle quite a few gene panel projects and exomes. Now, for those of you that do not have the hardware in place, there are other options. And more recently, DNASTAR has moved our software up to the Amazon cloud. And there's several advantages to using cloud hardware. One of them is that you don't have to invest and maintain your own hardware. Amazon will do that investment for you, maintain it, and it's always there. So the way that we use this then is we use our laptop that's on the ground, and we use a DNASTAR protocol to upload the data to the cloud. The SeqMan engine assembler is up in the cloud, so we can run the assembly. Then we set it up on our local computer. The processing power is used up in the cloud. The data can be stored up in the cloud as well. When it's done, we can return it back to our LAN computer and do the analysis. And so there's several advantages. One, I find myself using the cloud just out of convenience. It's more convenient for me to work on my laptop and launch assemblies on a big, powerful computer than it is to walk over to my big desktop computer in the other room and run assemblies. Other times, it may be that you're traveling or working with a collaborator, and physically you're in a different location. Well, your data is still central. You can launch assemblies. When you're traveling, in the hotel room, I can run assemblies if I forgot to run some, for a presentation. So the cloud option then has become a really nice option for our customers, and this is really gaining traction and acceptance as people are more comfortable putting data up on the cloud. We're definitely seeing a shift of people using this kind of new technology. Another major component to DNASTAR is the software support that we provide. And if you go to our website, under the support section, there's a number of different training videos. And I know the font is quite small here, but you can see there's a number of Ion Torrent and PacBio and Illumina training videos that are between 30 seconds and 5 minutes generally. And these are on specific topics, NGS topics and non-NGS topics for different molecular biology type workflows. So it's a great source. There's, I believe, over 200 videos now. They're also, you'll typically watch these through YouTube. So they're a great resource for our customers. Then we also do monthly webinars. And the webinars are on specific topics. I've done a couple of them this year, one with Ion Torrent data and Illumina, similar to this webinar for accuracy. I have one coming up later this month for microbial genome assembly. So the webinars are about an hour long. I spend some time introducing the software, but then the majority of the time is spent in the software with live software demos. So if there's a particular workflow that you're interested in addition to what we're talking about today, these webinars are archived back to 2012 on all different topics. So I highly recommend checking out some of the webinars if you're interested. So one of the first questions that we'll get with DNASTAR software is, can desktop software be powerful enough to handle large NGS data sets? And so I like to point people first to the assembly speeds, and these are some of the benchmarks that we've gotten, and these are actually going to get updated soon, but just to give you an idea, a whole human genome, which can be 1.2 billion reads in this case, almost 40x coverage, takes about, we'll call it 12 hours, 13 hours to both assemble and do the variant analysis. So we roll everything under the hood and do it together. We do the alignment plus the variant analysis. A lot of programs will kind of do those separately in separate steps. These times are going to improve quite a bit on some of the newer Mac computers in particular that have some really nice hardware components. These times are going to be dropping to about 8 to 9 hours. So we'll look for these to improve. Things like exomes, which are, of course, the large targeted resequencing, generally say about 2 hours. Gene panels, so an Ion Torrent panel or an Illumina panel, are typically measured in minutes. And so we get really, really fast assembly time. And we've had these assembly times for several years. Our algorithm is designed to be very efficient with large amounts of data. And really in the last year to year and a half, our focus has shifted more from speed and shifted to be the most accurate possible assembler. And so really the focus has been in that area now, and we've seen some nice improvements. Right now, our accuracy, if we're going to make kind of one general statement, this is with Illumina data, is about 99.7%. So that means out of 1,000 SNPs, there may be 3 that are missed, but we get 997 on average. And of course, it's easy to make a claim for accuracy. I mean, what does that mean exactly? There's lots of different ways that you can make an accuracy claim. And we didn't think that was a good enough thing for our customers just to make kind of a simplistic statement. And so we provided some tools within our software that allows customers to run their own data sets. It could be one that you've generated yourself. It could be one of the public data sets that you can download and run the data through the software. And then our software will automatically calculate the accuracy. It'll calculate things, and we'll go into more detail on this. so that you can really gain confidence in not only the software but your entire process. And we think that's something that's very, very important for anybody that's working with gene panels or variant detection, to really be able to validate that entire process. And so that's something that we're going to talk about today. So we provide this as, we call it a validation workflow. So for our panels, we have the Mendelian or germline workflow, we have a cancer somatic workflow, and we consider the validation workflow then to be kind of its own sub workflow. And so we set it up so that it's as easy as possible for a customer to come into the software and set this up. And sometimes they need a little help from us, and we can help them with the bed files and VCF files and get things set up. But you can really start to improve your own process when you have a means to validate it. And so here's a kind of a schematic for what we're doing. And let me try an arrow here maybe. Okay. And so there are some key components to doing this validation process. And one is that you need a region that defines what the targeted regions are. So typically it's a bed file. And in this case, we're using a bed file from NIST Genome in a Bottle. And NIST Genome in a Bottle bed file, put an arrow right there, is a high confidence region. It's out of NIST, and they sequence the human genome, and it's genome NA12878, the most sequenced and analyzed human genome that's out there. And they sequence it with Illumina and 454, and I think Solid and Ion Torrent, and found that not all the human genome can be called accurately, but the vast majority of it, we'll call it, say, 80% to 90%. And so they provide a region then of the human genome where it's high confidence. And then they also supply a VCF file of known variations that are in that region. So these variations have been confirmed using multiple different SNP detection methods, again, multiple different sequencing platforms. And so these are publicly available. So these purple blocks represent publicly available. This is the best one that's currently available. It is this NA12878 data set. We can plug these into the DNASTAR software. Or the green boxes represent, you may have your own targeted file. You may have a bed file that says, I know there's variations in this, in this panel. I've analyzed this maybe prior with Sanger data. And so I've got a list of 100 SNPs that I know are in this data set. I'm going to test your software to see if you catch all of them. So you can use either or, feed them in this pathway. And what we can see down here with the data set, we load the data set into the assembler, into the variant caller, into our variant annotator. And what we get in the end then, after the variant calls are made, we get in our ArrayStar piece accuracy calculations and a report that just tells us what was our false positive detection rate, how many positives did we detect, what was our false negative detection rate. So it does that for you, and that's really the cool part about this workflow, is that this is pretty hard to do on your own. Most customers, up until we've had this NIST data set, would really validate just on a handful of SNPs. It might just be, I've seen, where they have 10 SNPs and start adjusting parameters to try to improve detection on 10 SNPs, where you really want to do that when you've got tens of thousands or millions of variants to calibrate your process. And so there's a link. If you go to our website, you can see there's a link here to different articles. We have a Gen article that explains this in detail. We've got white papers that explain this. So I encourage you, if you're interested, check out our website. So the process then for doing a validation workflow starts with our assembly software, and that's our SeqMan engine. And SeqMan engine has a kind of a welcome page, and it's a wizard to really guide us through the project setup. And so it's a really easy to use interface that's put on top of a very, very powerful piece of assembly software. And we have different options. I could create a new project. If I was going to run this assembly on the cloud, it's really as easy as just clicking the option, putting your credentials in, and then everything else in the wizard is essentially the same, except that you're actually browsing to data that's up on a cloud computer rather than your local computer. Everything else is identical. So it's a really nice way to run software. So it just looks like this login here. So then when you're loading data, again, if it's cloud data, it'll use this data drive, or if it's if you're running this locally, it'll just be browsing your local folders. And so the typical input data are FASTQ files, and so they'll have a FASTQ or a text extension. If you have Ion Torrent data, it may be a BAM file extension. And so we can load then multiple FASTQ files per sample. You may also have multiple samples. So for example, if you have 100 exomes that you want to run in a certain time period, our interface will allow you to set up the assembly of all 100 exomes. If I do that on the cloud, the advantage that I didn't mention earlier is that with the Amazon hardware, you can run those 100 essentially consecutively. So if you have to get a lot of work done in a very short period of time, the cloud computer is a great choice. So if you procrastinated before a meeting and you've got a bunch of stuff to do, I found myself in a position, I need a bunch of data done by tomorrow. And so I can launch them on the cloud, go to where I need to, if I fly to my meeting, I can go and download the data from the hotel. So that's happened before, I'll admit that. But if you don't have a high throughput need, you can run one assembly at a time or run it locally on your ground computer. So again, that's what the cloud kind of interface looks like. So it's really similar to the rest of our interface. So now we're kind of looking at another snapshot from SeqMan Engine, the next step, which is input a template file. Most customers that are working with gene panels will be aligning to a human genome reference. On the DNASTAR website, we provide a number of different genome template packages. And these packages include things like the DB SNP build. So we combine the latest SNP information from DB SNP with the latest assembly, so the latest genome build, right? And then we package that up, and you can download that from our website. So we can see here, I'm going to go back to the highlighter. We can see here that we've got several human reference sequences, including a mitochondrial reference sequence, and then some other model organisms that you can download from our website. And so the template file then, in this case, is a genome template package. At this point, I also load in my bed file. So if I'm doing, again, targeted resequencing, I'm probably analyzing genes or exons from a gene subset. The bed file then, or manifest file, it's from Illumina, will define those regions of interest within the genome, right? So the alignment is still done to the whole genome. The analysis is limited to just those regions of interest. And so a little bit of, here's a little bit more detail out of what we can do with the genome in a bottle data. And that's, we can do an intersection, and we have several of these for some of the common panels that are used. And what it is that the NIST bed file is represented by this red line, and the human chromosome, the green. And the NIST bed file says that, well, about 80% of this chromosome 1 is high confidence. So areas at the telomeres or centromere, there may be some areas that you can't call SNPs accurately in those regions. But the gene panel bed file is just these few genes scattered across the chromosome. So we can also make an intersection bed file that says it's really just these areas where the two overlap that I'm interested in, that have SNPs that are verified of interest. And so we can limit then the regions intersection between the two to find, here are the highly confident SNP calls in my sample. And so this is something, again, that we can help customers with. They want to make an intersection file like this with genome in a bottle to really get tight, accurate validations. So the input sequence files, if it's from the cloud, we load them in. If it's from a local computer, we can load unpaired or paired reads. There's multiple sample data handling up to the top. I can run them as separate projects typically. So again, it could be a large number of exomes, or it could be a number of gene panels. It's an easy setup. For the validation workflow, we also load the VCF file that contains all the confirmed variant positions. So this is essentially the answer. So we're doing this alignment, And there's a set of SNPs in this case that we know are there from some other means, whether it's from Genome in a Bottle or our own means. We say, load the VCF file, it's a control, and it's a validation type. So this is a very important piece. If you're going to do a validation, you need to essentially have the answer for that validation step. But it's built into our software, so it's nice and easy to use. So once you've done that, you can run the assembly, and then we'll get some output that we can analyze in the DNASTAR software. But one of the, I think, very important points is to discuss that we're not just doing SNP calling accuracy, that because DNASTAR software is producing a full assembly and starting with raw data and bed files and VCF files, we can do quality control checks across the entire gene panel. So we can do things like measure the coverage across the targeted regions. And so the coverage then is not always 100%. So if we're doing an exome, even the very best exomes will have about 90, we'll call it 95 to 96% coverage. All regions aren't captured. So we want to know if we're doing especially a custom panel, or if it's the first time that you're doing an exome, you want to know, did I get enough coverage, or is it insufficient? So we generate reports and visualizations so that you can easily navigate to those areas that were missed in the targeting. Or if you're designing primers or probes to capture new exons and you're adding them to your own custom panel, you really need this information to know, are you getting sufficient coverage? And then also the specificity of the coverage is, if I design a primer or probe to capture an exon, how specific is it? Am I grabbing pseudogenes that are closely related, or gene family members? And so that leads to overall accuracy. If we have a lot of loss in the coverage or specificity, our overall accuracy is going to be lower. The mapping efficiency really is the DNASTAR assembler in this case. So how well does SeqMan engine map the reads, align the reads, what's the accuracy there? The variant filtering then also has an impact. How well you filter will have a significant impact on the overall. And so all of these together combine for really your variant call accuracy. And so I'll show you some of the interface that we provide that makes this a lot easier to do and gives you a nice kind of visual to look at each one of these steps. So one of the first places that I would go if I do my assembly in SeqMan engine, we've got a couple places to do analysis, and one of them is called our SeqMan Pro. And SeqMan Pro provides all different ways to look at the assembled data. And it provides multiple different reports. So at the top here, we've got a report that is called the coverage of target region for NA12878. This is constructed off the bed file that we provided. So this is really a visual report then of that bed file. And we can see highlighted here, we've got contig ID, chromosome 1, the feature, which is the gene type, the location right here, this column. Those are the coordinates within that bed file that were targeted. Then we get a length of that region, then the depth of coverage across that region. And so you can see right at the top there's one where the coverage depth is only 0.81, and it's only covered 63.65%. So that might be one that we're interested in looking at to say, well, the rest of them are 100% coverage. Maybe the depth isn't quite as good. I might look at some of those. We do a normalized kind of value on how deep that coverage is. And so this report then is interactive. I can click on it on areas that might be of concern, and then I can get these other views. So one view is the strategy kind of a view that shows depth of coverage plots. And here you can see these red peaks where the coverage is over 200 over the top of exons. So these green arrows are CDS features and exons within the CDS. So we get this strategy view. We also get a sequence alignment view. We can go and look at the aligned data, and they're all interactive with each other. So it's really the best interface for analyzing in detail an assembly of data. There's also we call it an enrichment report, and so it's another report then. It's under the same tab, that includes things like, in this case, not targeted areas, coverage reports. And so the not targeted area can also be a concern. So what this means is, where did reads align outside of my regions of interest? So if you targeted, a set of genes, ideally every single read would map just to that limited area. In reality, though, the specificity of the targeting isn't 100%, and so you may get significant, depending on how the primers and probes are designed, you may get significant data aligning outside that region of interest. And here we can see, just by the feature list, that there's a lot of pseudogenes. So what's happening is that the targeting is not 100% specific. Data is aligning back to the pseudogenes, and we can see in some cases, it's significant. There's depth. This is 19,000x coverage on these pseudogenes. So again, you'll see this with many different data sets, but you want to interrogate that and see, maybe I have to redesign a primer or probe because I'm capturing too much pseudogene for that primer or probe. So it's a great report. The enrichment report then is a summary. It's another tab on the same. And the enrichment report then tells me things like the number of reads that align, the number of reads that align to the targeted areas. So we get a read enrichment, total number of bases. Then we get target coverage at different depths of coverage. One of the more common questions that I get regarding SNP analysis is, what depth filter should I use? It's kind of an unknown. There's some people say, well, limit it to 20x. Some people say, well, 10x is good enough. And then you'll hear others that'll say, well, 6 or 7x is actually good enough depth of coverage. And so that's a nice thing about the validation process, is we can give you accuracy assessments at a variety of depths of coverages. And so you can select based on real data what the best level of coverage is, not just on what might be good or what sounds good. So when the validation process is run, there's different accuracy metrics that are calculated at each one of the variant positions that are listed in the VCF file. And so again, this is the numbers that are automatically calculated in our software, and it includes things like true positives, false positives, true negatives, false negatives, FDR, and from those metrics we can calculate sensitivity and specificity, right? So these are documented again in our software, so you can understand exactly what they're measuring. So again, with gene panels, it's difficult, and we'll look at some of the raw data, but it's difficult to give one metric. In our marketing, we like to say, well, accuracy is 99.7, but what does that really mean when there's a balance between, for example, getting the absolute highest number of true positives, that gets balanced with how many false positives are in the data, as well as false negatives. And so we'll look at some of that kind of raw data to give you an idea what that means. So again, this is from a white paper that DNASTAR published late last year, or I forget if it was early this year, late last year, where we have some accuracy results. And the top table is some of the first data that we worked with, and it's the accuracy results for the NA12878 exome. And what we did is we decided, well, we have to pick a software that we know is really good. Don't pick on the softwares we know aren't so good. Let's pick on the best one. And so we decided to compare ourselves to the BWA mapper and the GATK genotyper, or the haplotype-based caller. So we use both of their different SNP callers. This is the software that's used with the Illumina pipeline. The mapper is separate from the SNP caller. And you need quite a bit of bioinformatics experience to run both of them. But once you do, you can get very good numbers. And then we compared them to SeqMan engine. And what we found now this is the exome called the Garvan from the Garvan Institute, so you can get access to this if needed. And you can see that BWA GATK does a very good job. So if you look at true positives detected, or maybe we'll go right to sensitivity, you can see that they're all above 99%, right? So 99%. SeqMan engine is about 0.5% better. And so when you dig into that to see what does that actually mean, you'll see that the true positives detected are all over 14,000. SeqMan engine picks up, about 100 or so more true positives. False positives, SeqMan engine picks up just a few more, so about 25 to 30 more. But the false negatives were much lower. And false negatives are generally what you want to avoid and keep that as low as possible. So BWA GTK is around 120 to 130. SeqMan engine is nice and low at 45. And so we think we have a nice combination of getting the most true positives, the fewest false negatives, which results in the best sensitivity. And if you look at the far right, the elapsed time, we're at least 3 times faster than BWA GTK. So we're a lot faster, a little bit more accurate than what we think is the best variant calling software. So we think we have them beat on both accounts. Now, someone might ask, well, how do you account for the difference? And there is some indication. We don't know exactly because it's hard to look at every position, to figure that out. But what stands out here is this bases covered in the bed interval. And what it looks like when we compare these two is that our mapper aligns to about 100,000 more positions. So we think that our aligner is doing a little bit better job than BWA. It's faster, it aligns more reads, gives us a better result. So that's how we interpret that. If you look at another data set, which is a gene panel from the same genome, so this is a smaller data set. The number of true positives here is, measured, it's a little over 700. We see the similar kind of a pattern. We detect a few more true positives, a few more false positives, but only a couple of percent more. Less false negatives again. We get 0 in this panel. On BWA, GTK gets 7, and that gives us a better sensitivity, right? And again, we're about 3 times faster. So we thought that was, again, comparing ourselves against what we think was the best competition. We think that we had been able to outperform. We can also say this is more recent. So this is not published yet. You'll see this coming out from DNASTAR in the next week or so, I imagine. So we said, well, let's see how we compare against a commercial competitor that is doing variant analysis. So we looked at SeqMan engine as compared to CLC Bio. And you'll see the numbers are a little bit different. True positives, we can see that CLC Bio detects about 300 more true positives. Then we look at the numbers a little bit more. The false positive rate is much higher with CLC Bio, which is a bit interesting. And probably more concerning, the false negative, this is 5 times more false negatives, and it leads to a really high FDR, false discovery rate, for the CLC software. And that, again, results in a lower sensitivity. And it's somewhat how the software is balanced. There is a balance. If you have the software set to detect as many true positives as possible, without the proper filtering, you can have a high false positive and more alarming, a higher false negative discovery rate. And so we think we do a good job in comparison with CLC Bio. The speed differences aren't as great. We are still faster. So again, we think the pattern holds true. We have the fastest assembler and the most sensitive. And we see similar results again with the gene panel. In this case, our software finds more true positives. Again, many fewer false negatives. 0 in this case. CLC is picking up 7. False discovery rates and sensitivity, we have them beat. So we like the pattern that we're seeing. We're continuing to try to use these validated data sets to improve the process. It's the only way that you can improve software and SNP calling is having large validated data sets to do this. I've actually run some more assemblies. So this was the data sets that we looked at exome wise are the Garvan Institute. There's also a couple more exomes from Eric Labs and the Icon Institute, Mount Sinai. These are on the GetRM website. And these are some additional NA12878 exome data sets that are publicly available, and you can run these through the validation process. And I did this relatively recently just to see how consistent are the numbers. And what we actually find is that the exome that we chose for our marketing materials probably didn't give us the best results. We can actually get better results with some of the other exomes that are out there. And in particular, the Mount Sinai exome has 35,000 variants in it. We have 99.92% sensitivity, and the bed coverage is 97%. So it's actually a really, really nice, strong numbers with these other exomes, and very consistent. And of course, we're finding more data sets are being published and being made available. We'll continue to use these data sets to improve our process. So some of the things that are coming for version 13, there's a lot of work again on further accuracy improvements. We've been focused on not only on SNPs, but structural variations, smaller indels. Those can be a little more challenging to detect accurately, so we're making improvements there. We're also working with enhanced somatic variation detection. There aren't quite the validated data sets for somatic variations. I know the Genome in a Bottle folks and the FDA are interested in having these developed so that we can test some of the somatic variant detection algorithms. So we're going to continue to do work there. Hopefully we get a good benchmark where we can really fine-tune the detection of rare variants. We're looking to streamline workflows in the future. It's kind of a continual process. Additional white papers, the CLC Bio white paper will be coming out in the next week or so. And then again, there's going to be some database tools and enhanced database analysis coming out in our version 13 software. So those are all things that we'll have more information on. So if you're interested, please get in touch with DNASTAR. So with that, I'll take any questions. Again, thank you for your time today, and I'll stick around here to answer any questions that come up. Thank you. Let's see. Oh, so I have a question here from Tammy from the University of Toledo, and she asks, what are your thoughts on variant calling on genes where the molecular functions have not been determined and only implied? Very good question. The focus today, of course, is on, we look at human data sets. DNASTAR software can work with non-model organisms. And so if you have say a partially annotated genome, you can use that as a template. You can create your own VCF files in our software. So if you've got, you may have a list of VCF files for your organism, you can use those. You can create new VCF files. You can do the full analysis in a non-model organism. The only thing that you wouldn't be able to do is the functional analysis. So if you do the SNP analysis and get a gene set, then you may have a lot of putative function genes in your analysis. So we have full functionality with the non-model or partially annotated genomes. Okay, well, if there aren't any other questions, the DNASTAR booth, we have people answering questions there, so please feel free to stop by. And again, thank you for your time, and goodbye. Oh, so I have, so Tammy has another question. For variant calling in human samples, seems like it's critical to know the function. Yes, and so today's focus was really on validation. There is downstream analysis in our software that can link functional implications. So we can pull in database information, link it up with the SNPs at both the SNP or the gene level, and do things like ontology comparisons. And yeah, so there are extensive downstream tools. We just didn't cover them today. Okay, thanks again for joining today. And if you do have more questions, I'll be happy to answer them from the DNASTAR booth. And goodbye.